Hey, before we start the video, if you like this sort of content and you want more, come check out Med School Bootcamp. We have content for Step 1, Step 2, Anatomy, and OMM. Right now, we're about to watch the Biochemistry section. And if you're looking for downloadable PDFs, along with shorter videos and quizzes after each video to help you reinforce the information, then you can find it all on Med School Bootcamp. We also have a Step 1 question bank with some of the most representative questions to help you prepare for Step 1. You can also create a custom Step 1 study schedule when it's time for dedicated. So if you're looking to crush it in med school this year and pass Step 1, come check out Med School Bootcamp. It's going to be your first link down in the description. Now on to the video.
Hi everyone, my name is Paige and thank you so much for joining me for a chapter on glycogen. We've got a lot to cover here. We're going to start by talking about glycogenesis and glycogenolysis, right? The building up and breaking down of glycogen, which is this storage form of glucose. We're going to talk a little bit about the regulation of those processes, and then we're going to get into the meat of the chapter here, which is all of these glycogen storage diseases. We'll give you an intro, we'll run through all of the big ones that you'll need to know, and then we'll do a summary at the end, just in case your exam is tomorrow. So let's get into it. So glycogen is the storage form of glucose in your body. It's stored as this big, long polymer of glucose, and it is released when your blood sugar is low. So let's take a look at what glycogen looks like with our image over here. And you'll recognize these rings of glucose right here that are being strung together. And as you can see, they're strung together linearly, and then there are branches coming off of that main line. We have different names for these linear bonds versus these sort of branch point bonds here. When we have a linear bond, we're going to call those alpha 1,4 bonds. And when we have a branch point, we're going to call that an alpha 1,6 bond. And you just want to be familiar with that nomenclature here. You don't have to be able to count carbons around the ring or anything like that. Just be aware that linear is 1,4 and a branch is 1,6. And the reason that we have this branching is because that is going to increase the solubility of this otherwise very long, very bulky, insoluble polymer. And as you'll see, putting together those branch points is a really, really important process of glycogenesis. And most of the glycogen in your body is going to be stored in two places. It's going to be stored in the muscle and in the liver. So let's get started by talking about glycogenesis, how we're going to build up these glycogen polymers. And let's take a look at our diagram over here. So just to get yourself oriented, over here we're looking at glycolysis, the process we've talked about a million times, starting with glucose and working our way down. In the light pink here, we're going to be talking about glycogenesis, and then we're going to show glycogenolysis also here on the same diagram. Now, this type of diagram in particular, I know can give students a lot of stress. I know I very much did not like picking apart this kind of diagram, but what I tried to do on this diagram is if there was a relevant visual representation of the words here of this flowchart, I would try and show that here with this dotted line pointing to the visual representation of the polymer and what's happening with the polymer. So anytime you see a line here like this, that's just showing you visually what's happening here with each of these respective words. So since glycogen is the storage form of glucose, we better be starting with glucose, and indeed that is what we are going to be doing. And then via enzyme 1 here in the liver, which is glucokinase, we're going to be turning that glucose into glucose 6 phosphate. And that's all stuff you've seen before. Another thing you've seen before is this next step here, which turns glucose 6 phosphate reversibly, but in this case we're doing the forward reaction, into glucose 1 phosphate. And glucose 1 phosphate is going to be a really important molecule throughout this whole process. And the enzyme that's going to run that is called phosphoglucomutase. Our next step here is going to involve enzyme 3 called UDP glucose pyrophosphorylase, and that'll turn that G1P into a UDP glucose. And if all of these steps are kind of looking loosely familiar to you, we've actually talked about them before when we talked about galactose metabolism. So now we have our galactose metabolism image, and all of these key players are still here on the map, right? We've got glucose and glucose 6 phosphate, and then glucose 1 phosphate can be created from glucose 6 phosphate via that enzyme phosphoglucomutase. And then you remember the next step we talked about is that formation of UDP glucose. We talked about the formation of UDP glucose in a slightly different way when we talked about galactose metabolism as well. All that's to say, if you're getting deja vu, you should be. You have seen some of these steps before. All right, so we turned glucose into G6P, into G1P, into UDP glucose. Now we're going to turn that UDP glucose into the linear form of glycogen. The enzyme that's going to run that is a very important enzyme here, enzyme 4 called glycogen synthase. And this is the rate limiting step here. And this step forms that linear glycogen chain. So those alpha 1,4 bonds here are being created by glycogen synthase. So we made that 1,4 bonded chain using glycogen synthase. Now we're going to make those 1,6 bonded branches that come off the chain using enzyme 5 here, conveniently called branching enzyme. And you can see that depicted here visually as well. I highlighted the branch here just so you can see what this enzyme particularly did. It added that branch onto the linear chain. And now that we've got those branches, we've got that soluble storage form of glycogen that we're ready to store until we need it. So let's go back to the outline, start back again with glucose, run through this one more time and fill in some gaps. So glucose turned into G6P via glucokinase. You've seen that before. That's just the first step of glycolysis. But then instead of continuing to push that G6P down through glycolysis, we took a little detour over here, turned that G6P into glucose 1-phosphate, and the enzyme that did that was phosphoglucomutase. And remember, that was a reversible reaction, as we kind of saw in the galactose metabolism video. We turned that glucose 1-phosphate into UDP glucose via the enzyme UDP glucose pyrophosphorylase. And remember, we had also seen that UDP glucose before when we talked about galactose metabolism. Then we took that UDP glucose and turned it into linear glycogen, and the enzyme that did that was glycogen synthase. And remember, I said that that is the rate limiting step, and since that is the rate limiting step, that is going to be the step that is the most easily regulatable in this process. And we're going to talk a lot about the regulation of glycogenesis as well as glycogenolysis in a future video, but for now, just understand that glycogenesis is going to be increased, so glycogen synthase is going to be increased when you are in the fed state, right? When you don't need to be breaking down glycogen to increase your blood sugar. Your blood sugar's fine, we have plenty of sugar, let's go ahead and store it away and make some glycogen. So it should make sense then that glycogen synthase, which is going to synthesize glycogen and store glucose away, its activity is going to be increased by insulin, which is released when you are fed, as well as G6P, which is going to indicate that a big bolus of glucose just came in, right? And it was broken down into G6P. That G6P will then be pushed along through the glycogen synthesis pathway. And then cortisol is also going to upregulate this enzyme. And that can get pretty complicated. I would just memorize that. And then using that same logic, you should be able to understand how glycogen synthase would be downregulated by glucagon, which is released in the starving state, as well as by epinephrine. Both of those are trying to pump up your blood sugar. So those are going to favor glycogenolysis instead. And then we talked about forming those alpha 1,6 bonds in our last step to form that final form of glycogen, and the very conveniently named branching enzyme is going to be doing that, and that brings us to the end of our glycogenesis pathway. So to recap what we've done so far, we talked about the structure of glycogen, which is the glucose storage polymer. We've got linear alpha 1,4 bonds, and then branch points that come off alpha 1,6 bonds that promote solubility. We create glycogen in a process called glycogenesis in the fed state as signaled by insulin, G6P, as well as cortisol. All of these are going to promote the breakdown of glucose through this pathway here that we discussed, eventually to linear and then branched glycogen. Now let's move on and talk about the opposite scenario, glycogenolysis, breaking down glycogen. All right, so we just finished talking about glycogenesis, making glycogen. Now we're going to be talking about glycogenolysis, when we break down glycogen, when we need to increase our blood sugar. Let's jump straight into the diagram first. It's a little easier to do this visually, and then we'll come back to the outline. So enzymes 1 through 5 on this diagram here, we're talking about glycogenesis. Now we're going to be talking about enzymes 6 through 10 here, glycogenolysis. We're going to start right here with our final branched glycogen that we just finished making, and it looked a little something like this. And the first enzyme we're going to look at is enzyme 6 here, which is glycogen phosphorylase. Glycogen phosphorylase is the rate limiting step of glycogenolysis. So this is also the step that's going to be hormonally regulated, and we'll talk about that when we get back to the outline. So what glycogen phosphorylase is going to do is it is going to insert a phosphate in between each of these bonds on the glycogen chain, specifically right here on the branch. So it's going to break these bonds apart by stuffing a phosphate in there. And then as a result, all of those glucose 1-phosphates are going to pop off of the chain. So it will insert the phosphate, and then that whole unit will peel off in the form of glucose 1-phosphate. So glycogen phosphorylase is going to keep doing that until there are exactly 4 glucoses left on the chain. So I drew too many before. But you can see in this case, we had a branch with 6 on it, and glycogen phosphorylase is going to peel off 2 of them, leaving us with 4 remaining. This highlight here is just showing you what changed in between these 2 steps, which is that these 2 G1P units popped off. When we have 4 glucoses left on the chain, that is called a limit dextrin. I like to think of it like we reached the limit of what glycogen phosphorylase can do. And before we continue on talking about what those limit dextrins do, let's talk about that G1P. Because these G1Ps are at the heart of what we're trying to do. We're trying to release those G1Ps. Why? Because G1P, and you can see the same symbol over here, can be turned into G6P by that reversible enzyme phosphoglucomutase, and then enzyme 10 glucose 6-phosphatase, or G6Pase, can turn that G6P into glucose. And that was the whole thing we were trying to do, was we were trying to make glucose to raise our blood sugar. So we had our big long glycogen chain right here, and then we plucked off a bunch of those G1P units, which are effectively phosphorylated glucose at the one position. We shuffled that phosphate around, and then we just plucked it off, and we were left with glucose. All right, so that was the fate of all of these G1Ps that we cleaved off. Let's get back to our limit dextrin now, which remember was that glycogen chain with 4 glucoses left on that branch. That was after we had cleaved off all of the rest of those in the form of G1P. Our next 2 enzymes, enzyme 7 and enzyme 8 here, are going to be called debranching enzymes. Enzyme 7 is called 4-alpha-D glucanotransferase, and what that's going to do is that's going to take that limit dextrin, it's going to take 3 out of those 4 glucoses that were remaining, and it's going to pop them right onto the end of the linear chain. So you can see now, off of that branch, we took 3 out of those 4 remaining glucoses, and we just kind of stuck them on the end of the chain we were already working with. Then the other debranching enzyme, enzyme 8 here, alpha-1,6-glucosidase, is going to deal with that last remaining glucose on the chain here. And it's just going to pluck that off. And that's just going to leave in the form of glucose. So remember, with glycogen phosphorylase, it was stuffing phosphate groups into the bond to get it to pop off, so then we had to modify it afterwards to turn it just into glucose. Here, the debranching enzyme doesn't need to stuff a phosphate in there, so it's just going to liberate 1 free glucose, just that last one that was left on the chain. And so once that glucose leaves, we are left with this newly linear glycogen, which is 3 glucose units longer than the original linear chain was, and the branch has been decimated. And in the case of this chain here, we ended up with 3 glucose in total, right? 2 from the G1Ps that we got, and 1 from the glucose that came off at the very end. So you're always going to get 1 glucose at the very end from that debranching enzyme activity. The number of G1Ps you get that then turn into glucose is going to depend on the length of the chain. So if that branch chain was much longer than the one we had here, glycogen phosphorylase would just keep hacking away at it until it got to 4 glucose left on the chain, until it got to that limit dextrin. One final enzyme to mention here is enzyme 9 here, which we haven't talked about yet, called alpha-1,4-glucosidase. This is specific to the lysosome. It is by no means a major glycogenolysis pathway, but it is implicated in glycogen storage diseases, so it is important to talk about. In the lysosome, alpha-1,4-glucosidase is just going to take that branch glycogen and turn it directly into glucose. Nice little shortcut there. And it would be so easy if it just worked like that all around. So that is a minor pathway, but an important one to be aware of. It'll come back up later on in the chapter. All right, so everything we've been talking about right now is glycogenolysis, and specifically, this is the pathway that's happening within hepatocytes. Don't worry, it's very similar for skeletal muscle. There is just a bit of a nuance there. So for hepatocyte glycogenolysis, remember the first step there was glycogen phosphorylase. That was the rate-limiting enzyme, and it has a vitamin B6 cofactor, which is important to be aware of. Remember, that was stuffing phosphate groups, or phosphorylating those 1,4 bonds and cleaving off G1P units until we reached 4 glucoses left on that branch there. And once we reached the form of glycogen where there were 4 glucose units left on the branch, that was called the limit dextrin. Remember, those G1Ps were useful because we could turn them into G6Ps via that enzyme phosphoglucomutase. And those G6Ps could be acted upon by G6Pase, the enzyme, in the liver and the kidneys to form glucose, which is the whole point of this whole thing is raising the blood sugar. Notice I said the liver and the kidneys, and what I did not say is the muscle. That's the nuance to the skeletal muscle discussion that we're going to get to in a second, so put a pin in that for now. And like I said, the step mediated by glycogen phosphorylase is the rate-limiting step. It's the step that is going to be regulated by hormones, and its activity is going to be increased anytime we're trying to increase that blood sugar. So epinephrine and glucagon are both going to increase its activity, and insulin is going to decrease its activity. Glycogen phosphorylase activity is independent of the ratio of AMP to ATP. And that's because the liver is in charge of increasing the blood sugar around for the rest of the body, okay? The reason I mention this here is because that's not going to be true for skeletal muscle. We're only going to be regulated by hormones in the liver. All right, so trucking on down the rest of the pathway, we're getting back to that limit dextrin we were talking about. And the next thing we were going to do is take 3 out of those 4 remaining glucose on the chain, and we were going to move them from the branch to the linear chain. Our first debranching enzyme, called 4-alpha-D-glucanotransferase debranching enzyme, is going to be in charge of that step, and then that last glucose on the branch is going to be cleaved by the next debranching enzyme, alpha-1,6-glucosidase. And that just makes a unit of free glucose that goes off into the blood. And we already talked about why the direct production of G1P is so much higher than it is of glucose in this process, because only that last glucose unit on the branch is being cleaved off just as glucose. All of the rest of them have to come off as G1Ps that then will turn into glucose after later modification. And then we talked about that special pathway in the lysosome where some of that glycogen can just be degraded by alpha-1,4-glucosidase directly into glucose. All right, so let's talk about skeletal muscle now. Almost all of those steps that we just finished talking about are going to be exactly the same, with a couple of key differences. The first key difference is that glycogen phosphorylase, in addition to being regulated by epinephrine and insulin, like we talked about, are going to be regulated by a few more things allosterically. So in addition to being hormonally regulated, skeletal muscle will also be allosterically regulated by some energy production products. So its activity is going to be increased by AMP and calcium in addition to epinephrine, and decreased by ATP in addition to insulin. AMP is a signal of a low energy state in the muscle, and calcium is a signal of muscle contraction. Both of those are signaling that the muscles are working really hard. We should increase the sugar because clearly these muscles need it. Just like in our other pathway, we're going to be creating G1P from glycogen via phosphorylase and the debranching enzymes. None of that is new. That G1P will turn into G6P via phosphoglucomutase. Again, you've seen that before. But the difference is in skeletal muscle, enzyme 10 here, G6Pase, does not exist. So we turn G1P into G6P, but we're not going to turn that G6P into glucose. Instead, that G6P is immediately going to be shuttled down through the rest of glycolysis and burned for energy. So come take a look at the diagram here. If we don't have enzyme 10 turning G6P into glucose, G6P instead can just run down through the rest of glycolysis, but it's not going to turn straight into glucose. The reason is because skeletal muscle is selfish. It's not like the liver, right? The liver is trying to put glucose into the blood for all the other organs to use. Skeletal muscle doesn't work like that. Skeletal muscle is looking out for number one, and it's only going to break down glycogen when the muscle needs it for energy. So it's not going to send glucose floating around in the blood. It's going to make G6P and then immediately snatch that G6P into the glycolysis pathway and just use it right there, right away. Like I said, that muscle phosphorylase is dependent on the AMP to ATP ratio, right? That's an indicator of its own energy supply. Muscle is only storing glycogen for its own consumption. It's not looking out for any of the other organs in the body, so no need to turn it right into glucose. So that's the difference between skeletal muscle glycogenolysis and hepatocyte glycogenolysis. So let's talk about everything we've talked about across glycogenesis and glycogenolysis. Let's wrap it up. Glycogen, a storage glucose polymer stored in your body for when your blood sugar gets low. It's characterized by a branched structure. So there will be linear alpha-1,4 bonds and then branches coming off of those linear chains, making 1,6 bonds. And the reason that we have those branches is to increase the solubility of that glycogen, which is stored in the muscle and the liver. To create glycogen and put it into storage, we're going to start with glucose. We're going to do the first step of glycolysis, which is turning it into G6P. And then instead of continuing down glycolysis, we go on a little side quest here, turn that G6P into G1P, follow a couple more steps here, eventually get a linear form of glycogen and then a branched form of glycogen, which is our final form. The rate limiting step of glycogenesis was glycogen synthase, this enzyme here, which is increased in the fed state. So any hormones or molecules that are signaling a fed state, and then any hormones or molecules that are indicating that our blood sugar is low or we would need to increase our blood sugar, we're going to shut down glycogen synthase. Instead, when that blood sugar is low, we're going to undergo glycogenolysis. Rate limiting step here is glycogen phosphorylase, which we said was going to phosphorylate and stuff phosphates into each of those bonds on the branch, picking off glucose 1-phosphate units until we leave 4 glucose left on the chain. We called that a limit dextrin. And then we did a couple more modifications basically to move these glucose units around and end up with one fully linear chain where we ended up getting rid of the branch point in question. And we picked off some glucose, and like I mentioned, those G1P units as well. Those G1P units in the liver could turn into glucose via G6Pase, but that was not the case in the muscle. In the muscle, there was no G6Pase. Instead, once we had that G1P, we turned it to G6P and then just sent it right back down for glycolysis for immediate consumption by the muscle. That was because the muscle is looking out for number one and is regulated by its own energy supply in addition to hormones. And stay tuned for the next video where we'll talk about all of the different types of regulation of glycogenesis and glycogenolysis by hormones and allosteric molecules. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on the regulation of glycogen metabolism. So we just finished talking about glycogenesis and glycogenolysis, and we started to talk a little bit about how both of those processes are regulated both in the liver and in the skeletal muscle. And in this video, we're going to unpack some of those signaling pathways a little bit more and really hammer home those points. So our key hormonal players that we discussed in the last video that are going to be regulating glycogen metabolism were insulin, glucagon, and epinephrine. Remember, we also talked about how in addition to hormonal regulation, skeletal muscle had a special situation where it was also being regulated allosterically by its own energy supply. So those were molecules like AMP, ATP, and calcium from the contraction of skeletal muscle. And the reason that this was happening is because skeletal muscle is storing glycogen only for its own use. We talked about 2 rate-limiting enzymes in the last couple of videos. For glycogenesis, we said that the rate-limiting enzyme was glycogen synthase. And for glycogenolysis, we had identified glycogen phosphorylase as that rate-limiting step. One high-yield point that I want you to remember that's going to help you make sense of a lot of these different pathways is that phosphate is going to signal starvation. What I mean by that is that when you are starving, when your body's breaking down a ton of ATP, you have a lot of phosphate groups floating around. It's a signal of a low energy state. That's going to be helpful to remember because in the starving state, your body is going to want to increase blood sugar. That means it is going to activate enzymes like glycogen phosphorylase, which are glycogenolytic. The phosphorylated form of those enzymes are going to be the active form. Okay, so any enzyme that is raising your blood sugar that we're going to be talking about in this video is going to be active in its phosphorylated form. And you'll see that on our diagram over here as well. Alternatively, enzymes that are active in the fed state, like glycogen synthase, are going to be deactivated in their phosphorylated form. So like I said, glycogen phosphorylase. In addition, there's an enzyme that phosphorylates glycogen phosphorylase. So it adds a phosphate group onto glycogen phosphorylase, therefore activating it. And that enzyme is called glycogen phosphorylase kinase. And just like glycogen phosphorylase has an activating enzyme, glycogen synthase also has an activating enzyme, and that is called protein phosphatase. And that is going to dephosphorylate and therefore activate glycogen synthase. So let's go ahead and talk about some of these hormones. We're going to start off by talking about glucagon. You've already seen this glucagon signaling pathway in part if you watch the regulation of phosphofructokinase video. The signaling pathway for glucagon is going to be active in the liver. It signals a starving state. So in the starving state, you should be thinking way downstream. Our end goal here is to increase the activity of glycogen phosphorylase to eventually make glucose. And we want to increase that activity by phosphorylating that enzyme. The enzyme that activates glycogen phosphorylase, remember, was glycogen phosphorylase kinase. And we want that enzyme to be phosphorylated as well so that it is active to phosphorylate our glycogen phosphorylase. How's it going to do that? Well, glucagon binds to a G protein coupled receptor, and it's going to activate the adenylyl cyclase pathway. So when glucagon binds that G protein coupled receptor, those subunits are going to pop off and they are going to activate adenylyl cyclase. And adenylyl cyclase is going to turn ATP into cyclic AMP. That cyclic AMP is going to go on to activate protein kinase A right here. And the job of protein kinase A is to just go around to a whole bunch of different cell signaling pathways and just slap phosphate groups on things, okay? That's, basically what it does. So one of the things it's going to slap a phosphate group on is glycogen phosphorylase kinase right here. So once pKa adds that phosphate group, we've now activated the glycogen phosphorylase kinase, which activates the glycogen phosphorylase, which breaks down glycogen into glucose. Okay, so let's go back to the outline, take a little bit of inventory. So remember, glucagon is released when the glucose is gone, and it's going to indicate the starved state. And this pathway is happening only in the liver, not in the skeletal muscle. So this is a catabolic pathway that is breaking down glycogen. Glucagon binds the G protein coupled receptor, which is going to activate the adenylyl cyclase, turns ATP into cyclic AMP, which activates protein kinase A. Protein kinase A starts flying around the cell, putting phosphates on things, including glycogen phosphorylase kinase, which is going to activate glycogen phosphorylase. The other thing that protein kinase A does in its efforts to put phosphate groups on everything is it is going to decrease the activity of glycogen synthase. Remember, phosphate is going to deactivate glycogen synthase. So when pKa goes over to glycogen synthase and tries to slap a phosphate group on it, that is going to deactivate that enzyme and favor glycogenolysis. All right, now let's talk about the opposite scenario. Now we're talking about the fed state, and we're going to talk about insulin. Insulin is released after you take in a big glucose-rich meal, and it is going to conduct its signaling pathway in the liver and in the muscle. And here it's going to have an anabolic effect on glycogen. So it's going to promote glycogenesis and decrease glycogenolysis. Now we're back to our diagram, and we're going to focus on our green insulin pathway here. One thing just to note about this diagram, anything in these warm red colors here are going to indicate the starving state. Anything in these cool and green colors here is going to indicate the fed state. All right, so now we're talking about insulin. We're talking about the fed state. Insulin is going to bind not a G protein coupled receptor, but a receptor tyrosine kinase, right? This right here. And since we already know that glucagon is going to increase cyclic AMP, it goes to reason that insulin is going to decrease cyclic AMP, because we don't have that glucagon signaling. So we don't have the cyclic AMP floating around, we don't have protein kinase A, and we're not phosphorylating all of these enzymes around the map. So now these enzymes are in their dephosphorylated state, which means that these fed state enzymes, now it's their time to shine. So we are going to have active protein phosphatase, which remember was the activating enzyme for glycogen synthase. And the purpose of glycogen synthase is breaking down glucose to put into storage as glycogen. And additionally, the binding of insulin to its receptor tyrosine kinase is also going to directly upregulate glycogen synthase activity. One thing also to mention about this protein phosphatase activating enzyme right here is it is a phosphatase, meaning it is going to cleave phosphate groups off of things. One of the things it's going to cleave phosphate groups off of is glycogen phosphorylase kinase. If we scrap that phosphate group, we're going to deactivate that enzyme, which deactivates glycogen phosphorylase as well. So let's take it back to the outline. So insulin binds the receptor tyrosine kinase, which decreases cyclic AMP, and is basically going to undo that whole glucagon pathway that we just finished explaining. Protein phosphatase gets upregulated, as does glycogen synthase. And remember that protein phosphatase also went over and sort of shut off that glucagon signaling pathway in a sense by decreasing glycogen phosphorylase kinase activity. And then we also talked about how insulin directly can increase the activity of glycogen synthase. The last hormone we're going to talk about is epinephrine. And epinephrine is going to signal to the body, like, we're doing something very extreme, we better make sure we have enough glucose in our bloodstream to handle all of this stress and excitement. So this is going to have a catabolic effect on glycogen, similar to glucagon. And there are two kind of signaling pathways to be mindful of here. The first one, which is happening in both the liver and the muscle, is happening when the beta adrenergic receptor is activated by epinephrine. And the second one is when the alpha adrenergic receptor is activated also by epinephrine. That's only happening in the liver. So let's start by talking about the beta adrenergic receptor, and that's going to do the exact same thing that glucagon did. It is going to activate adenylyl cyclase, turn ATP into cyclic AMP, and it's going to go down this whole pathway like we already talked about. It's the same. But this other pathway is happening only in the liver. That's going to trigger the IP3 DAG signaling pathway. And we're not going to get into all the nuts and bolts of the IP3 DAG pathway in this video. Just understand that it is going to signal via something called phospholipase C, and it is going to have activity at the endoplasmic reticulum and trigger the release of calcium. So we are going to increase intracellular calcium. That's kind of the upshot of all of this. This increased intracellular calcium not only is going to directly increase glycogen phosphorylase kinase, but it is also going to complex with calmodulin, and that is a signaling particle too that is going to contribute to the upregulation of glycogen phosphorylase kinase. So back to the outline, the beta adrenergic receptor active in the muscle and the liver is working the exact same way as glucagon. It's going to eventually downstream via the protein kinase A pathway upregulate glycogen phosphorylase, downregulate glycogen synthase. And then in the liver, we had the extra help via the alpha adrenergic receptor, which is going to activate the IP3 DAG pathway via phospholipase C. And that worked to increase intracellular calcium, and that all upregulates glycogen phosphorylase kinase as well, both directly and then also via the calcium calmodulin complexes. Then finally, remember we said there's this extra bonus mechanism in the skeletal muscle. It's going to be regulated by its own energy supply. So when there are signals of an increased need for glucose in skeletal muscle, we are going to promote glycogenolysis. So remember, those were AMP, which comes from the breakdown of ATP from using all that energy, as well as calcium from muscle contraction. When those muscles are contracting and we're increasing that intracellular calcium, that's going to have the same effect that that increased intracellular calcium had when we were talking about the alpha adrenergic signaling pathway in the liver. The calcium will bind with calmodulin that will promote the activity of glycogen phosphorylase kinase. By the same logic, then, signals of a high energy state in the muscle where we can go ahead and store some glycogen are going to be ATP and glucose 6-phosphate. ATP should be pretty self-explanatory, that is energy, and glucose 6-phosphate is just indicating that we have enough products of glycogen phosphorylase. So both of those signal a high energy state, promote glycogenesis. So let's wrap it up, shall we? There are two main buckets of regulation of glycogen metabolism. We've got hormones, and then we've got allosteric regulation, the latter of which is specific to skeletal muscle only. When we are in the starved state, we're looking for enzymes that are going to be activated by being phosphorylated. So those enzymes are glycogen phosphorylase as well as glycogen phosphorylase kinase. Alternatively, in the fed state, when we're feeling anabolic, we're looking for enzymes that are active in their dephosphorylated state. Those are going to be glycogen synthase and protein phosphatase, which activates it. Glucagon and epinephrine both worked by ultimately activating protein kinase A, and protein kinase A went around and put phosphates on all the things that needed phosphates and activated glycogen phosphorylase and glycogen phosphorylase kinase while shutting down glycogen synthase. Additionally, there was another epinephrine pathway that worked by increasing intracellular calcium, had a very similar effect in the starved state. In the fed state, we talked about insulin and how with insulin signaling, we weren't getting that increase in cyclic AMP, so we are going to deactivate all those glucagon signaling pathways. We were going to have a bunch of dephosphorylated enzymes, and that meant that it was time to shine for glycogen synthase as well as protein phosphatase, which activates it. Then finally, we talked about how in the skeletal muscle, signals of a low energy state like AMP or increased intracellular calcium, which signals muscle contraction that those muscles have been working, those were going to promote glycogenolysis, and the opposite was true when we had high energy signals like ATP and G6P. That's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our overview lesson on glycogen storage diseases. So we spent the beginning of the chapter talking about all of the different steps in glycogenesis and glycogenolysis. There were a lot of steps. As such, it's probably pretty easy for you to imagine all of the different ways that these can go wrong. And so when there is an inborn error in one of these glycogenesis or glycogenolysis enzymes, that is a glycogen storage disorder. It's an enzyme deficiency in either glycogenesis or glycogenolysis. And for the purpose of the step exam, they're going to want you to know types 1 through 6, all of which are inherited in an autosomal recessive fashion. And there are a couple of symptom buckets you can kind of think of these in to help you categorize them in your mind. They can affect the liver, they can affect the muscle, or they can affect both the liver and the muscle. And that should make sense to you, right? We talked about how glycogen is primarily stored in the liver and the muscle. And while the symptoms can all vary a lot between these diseases, one keyword that you really want to just keep your eyes peeled for, it'll point you in this direction, is a positive periodic acid shift stain. So periodic acid shift staining, or PAS or PAS, that is a stain that is going to stain polysaccharides like glycogen. And so a buildup of glycogen in affected tissue due to a defect in, say, glycogenolysis will cause a buildup of PAS-positive material in the affected tissue, either in the liver or the muscle or both. So I'm going to take you into our diagram here and just introduce you to types 1 through 6 of these glycogen storage disorders. We're going to have an individual video on each one of these disorders. I just want to introduce you to the full spectrum of them. Glycogen storage disease type 1, probably one of the most highly tested, is von Gierke disease. Von Gierke disease is a deficiency in glucose 6-phosphatase, or G6Pase. Remember, skeletal muscle does not have G6Pase, so this is primarily going to affect the liver. GSD type 2 was called Pompe disease. And remember how we talked about in the glycogenolysis video, this kind of random little pathway that only happens in the lysosome. It's not really a major pathway, but it is an important one, where glycogen is getting directly broken down into glucose. A defect in that enzyme, alpha-1,4-glucosidase, in the lysosome is going to cause Pompe disease, and Pompe disease, unlike von Gierke disease, is going to primarily affect the muscle and sometimes the liver. GSD type 3 is called Cori disease, and you'll see it affects 2 enzymes here. These 2 enzymes were what we lumped together and we called the debranching enzymes. That first enzyme was 4-alpha-D-gluconotransferase, and the second one was alpha-1,6-glucosidase, and together they helped move those glucose monomers off of the branch and onto the linear chain. That's going to affect both the muscle and the liver. So you're going to see symptoms in both kind of buckets. GSD4, it's called Anderson disease. And it's interesting because it's the only one of these GSDs that's actually taking place in the glycogenesis pathway. All of the others are a defect in glycogenolytic enzymes. And this is a defect in branching enzyme, which added the branches onto the linear glycogen chains. So, branching, debranching, it can get a little confusing. So you can remember A, B, C, D. Anderson is branching enzyme. Cori is debranching enzyme. Alternatively, you can remember that Anderson can't add branches, right? So the letter A, and Cori can't cleave branches, so the letter C. And Anderson disease, I should mention, affects the muscle and the liver, as does Cori disease. Finally, enzyme 6 here actually has 2 different glycogen storage diseases associated with it, depending on if you're talking about the muscle or the liver. That enzyme, of course, being glycogen phosphorylase, which cleaves the branches of glycogen down into the limit dextrins. The limit dextrins, remember, being the branched form of glycogen with 4 glucose monomers on the branch. When you're talking about muscle phosphorylase, you're talking about McArdle disease. Alternatively, if you're talking about glycogen phosphorylase in the liver, then you're talking about HERS disease. So GSDs 5 and 6 both affect the same enzyme, just in a different location. So now that you're familiar with sort of which steps are going awry and which GSDs they're corresponding to, let's go back to the outline and talk about sort of the muscle bucket versus the liver bucket that I was talking about before, so you can start to identify some symptoms and narrow things down. So when you have liver involvement with some of these glycogen storage diseases, some things that you might see on presentation include fasting hypoglycemia, ketosis, and hepatomegaly. Remember, that is in conjunction with that keyword that I pointed out to you, that periodic acid shift positive material on biopsy is like a dead giveaway. So in this case, you would see that on a liver biopsy. And so the 4 glycogen storage diseases that fit into this liver bucket, and so you might see some of those liver symptoms, are 1, 2, 3, 4, and 6. So like we discussed, type 1 is von Gierke disease. That is a problem with G6Pase, so cleaving that G6P into glucose. Type 3 was Cori disease, which was a defect in both of those debranching enzymes. Instead of debranching enzyme, if the problem is with branching enzyme, that was Anderson disease. And then of course, a problem with glycogen phosphorylase in the liver, that is Her's disease. Okay, the other bucket of symptoms, like we were discussing, was muscle involvement. And this can kind of be broken down into 2 other subsections. If the defect is in glycogenolysis, you will see exercise intolerance, cramps, easy fatigability, and rhabdomyolysis. So here you're thinking types 3 and type 5, right? Cori and McArdle. Now another symptom you might see is progressive weakness. And you're going to see this symptom if there's a defect in glycogenesis, like in Anderson disease, or lysosomal glycogenolysis. So remember, that was Pompe disease, that alpha-1,4-glucosidase. And muscle involvement might include cardiac muscle involvement. And you'll see cardiac muscle involvement as a possibility in a couple of these different disorders. The one you might want to really focus your attention on is type 2 Pompe disease. So like we said, the GSDs that are involving the muscle are going to be 2, 3, 4, and 5. Remember, 2, 3, and 4 also had liver involvement, so you're going to see them sort of with both presentations. And then types 2 and 5 are primarily the muscle. Type 2 being Pompe disease, which was a decrease in that lysosomal alpha-1,4-glucosidase enzyme, and type 5 being McArdle disease, which was a decrease in the muscle glycogen phosphorylase. So in summary then, a glycogen storage disease is a defect in an enzyme, either in glycogenesis or glycogenolysis. The best way to sort of start thinking about these in a question stem is to look for the presence of periodic acid shift stain positive material, either in the liver or in the muscle. If you see it in the liver, and you see liver involvement symptoms like fasting hypoglycemia, ketosis, hepatomegaly, and they don't have to have all of these, but these are good indicators that the liver is involved, you're thinking about GSDs 1, 3, 4, and 6, von Gierke disease, Cori disease, Anderson disease, and Her's disease, with defects in these corresponding enzymes here. And if you're seeing that past positive material in a muscle biopsy and you're seeing muscle symptoms like exercise intolerance, cramps, rhabdo, progressive weakness, easy fatigability, right? Now you're thinking muscle involvement, you should be thinking of 2, 3, 4, or 5. So that was a lot of information. We're going to do a deep dive into each of these disorders, so you'll start to be able to parse them out by the end. But for now, just sort of start to categorize them in this way, and you'll be in good shape. Otherwise, let's move on to our first glycogen storage disorder, von Gierke disease. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on von Gierke disease. For this next part of the chapter, we're going to be going into specific details about each of the 6 highest yield glycogen storage diseases that you might see on the step 1 exam. We're just going to go through them numerically. So glycogen storage disease type 1 is von Gierke disease. And all of these glycogen storage diseases, like we talked about in our overview video, are enzyme deficiencies either in glycogenesis or glycogenolysis. In the case of von Gierke disease, this enzyme deficiency, which we'll go into the pathophys of in a second, is going to be a defect in a glycogenolysis enzyme. However, a unique thing about the enzyme that's deficient in von Gierke disease is that it is also implicated in gluconeogenesis. So von Gierke disease is a little bit unique, and it's going to have a pretty severe presentation. And that defective enzyme that leads to impairment in both glycogenolysis and gluconeogenesis is going to have effects in the liver. So if you remember from our overview video, we sort of had 2 buckets of symptoms, right? A liver presentation and a muscle presentation, and you could have either or both of them. Here we're going to be focused on the liver bucket of symptoms for von Gierke disease. So what is exactly this defective enzyme in von Gierke disease? We've actually talked about it in another video. So here's our diagram of glycogen metabolism, and you can see von Gierke disease listed right here as a defect in enzyme 10 right here, enzyme 10 being glucose 6-phosphatase. And the purpose of glucose 6-phosphatase was turning glucose 6-phosphate into glucose. So let's cut over to our metabolic map just for a second to talk about the wider metabolic implications of a defect in G6Pase. Let's zoom in sort of up on the top of the map here. And so here you can see is G6P, here is glucose, which would mean that this step right here is going to be mediated by G6Pase, which is defective in von Gierke's disease. So if we knock out this step here, not only does it block the final transformation, turning G1P to G6P eventually into glucose in glycogenolysis, but as you can see, that step is also implicated in the gluconeogenesis pathway. If we follow gluconeogenesis all the way up, remember that last step is running glycolysis in reverse. It's the exact same step, turning G6P into glucose. So that means in patients with a G6Pase deficiency, right, with von Gierke disease, not only can they not break down storage glucose, glycogen, when they are hypoglycemic, they also can't undergo gluconeogenesis and make new glucose de novo. And you would imagine this is completely catastrophic for maintaining blood sugar levels. So von Gierke disease is a defect in G6Pase activity. Technically, there are two subtypes of von Gierke disease. The one that you are much more likely to be tested on is type 1A. And type 1A is a defect in the enzyme itself from defective enzyme expression. So the gene that mediates that expression is the G6PC gene on chromosome 17q. However, there is technically another subtype of this that will present the same way. Instead of being a decrease in the enzyme expression itself, this is going to be a decrease in the translocator protein. So if you have a gene defect in the SLC37A4 gene on chromosome 11q, you're going to get a decrease in that G6P translocase. And that's called type 1B von Gierke disease. That translocase is a G6P transporter protein that is present in the liver and the kidneys and the GI mucosa. And what it will do is it will bring that glucose 6-phosphate into the endoplasmic reticulum where the G6Pase enzyme is active for it to do its hydrolyzing of the G6P. So if you're deficient in that translocator, then the G6P can't get to the G6Pase, so it'll have the same effect as not having a functional G6Pase at all. Regardless of which mechanism the patient is affected by, in both cases, you're getting a decrease in the breakdown of G6P by that G6Pase enzyme. You're going to get a buildup of G6P and a decrease in glucose production from G6P. So let's move on now and talk about the presentation of von Gierke disease. Remember, we said that a G6Pase deficiency is going to fall under our liver bucket of symptoms. So the first thing you might be thinking of is hepatomegaly. In this case, we're also going to see renomegaly. And both of these are happening because of a buildup of glycogen in these cells as well as lipids. We already talked about why you get a buildup of glycogen, and with the lipids, we're going to be talking about that a little more once we get down to the diagnostics in terms of how that all happens, so just hang tight. Another presentation you might see are hepatic adenomas, and this is something you should just memorize. The pathophysiology of this is very poorly understood. Now, a very common chief complaint that you'll see in vignettes about von Gierke disease have to do with hypoglycemia. So you might see a tremor or confusion, or very frequently things will get bad enough that the patient will have a seizure, and that's what will bring them to your office. Another presentation might be gout, and that is because these patients suffer from hyperuricemia, which again we will talk about in a minute when we talk about diagnostics. And finally, maybe a lower yield presentation, but something that they might drop in the stem, so don't let it confuse you, is dull-like facies, or a round face from fat accumulation in the face. Now, let's move on to diagnostics, and the hallmark diagnostic of von Gierke disease is going to be severe fasting hypoglycemia with lactic acidosis. And that is because these patients are entirely reliant on fat and protein catabolism for their energy, because, right, they don't have any glycogen breakdown, and they don't have any gluconeogenesis. And that's why those chief complaints are usually something surrounding an acute hypoglycemic episode. These patients just cannot maintain their blood sugar. And that's a very high yield point in most of the questions you see surrounding von Gierke disease, and you do want to remember that lactic acidosis because that's going to be important when we come back and talk about our differential diagnosis. So another thing you might see on labs is hyperlipidemia with a mild ketosis. And the reason that that happens is because the hypoglycemia leads to a sympathetic activation, and that sympathetic activation is going to stimulate lipoprotein lipase. And if you're following this biochemistry unit chronologically, we haven't quite talked a whole lot about fatty acids yet. So for now, just understand that that upregulation in lipoprotein lipase is going to promote the release of free fatty acids. And then those those free fatty acids will go to the mitochondria and be oxidized into ketones. Now, like we alluded to earlier, these patients may suffer from hyperuricemia. The reason that this is happening is because that buildup of G6P is going to lead to a decrease in intrahepatic phosphate, right? Instead of having free glucose, we have our glucose with a phosphate still attached. So all of those phosphates are being consumed and being sort of sequestered in the form of this G6P. That decrease in intrahepatic phosphate is a signal to the body to do purine degradation. So the activity of AMP deaminase is going to be increased and purine degradation is going to be increased. And remember, that is the whole mechanism of hyperuricemia. Another thing that might be described, since we're thinking about our liver bucket of symptoms for von Gierke disease, is an increase in liver function tests. So they might describe an increase in serum biotinidase. It's a marker of liver function, similar to these liver function tests like ALT, AST, and ALK-Phos. These patients might also have liver cirrhosis. So on an ultrasound, maybe they'll describe a nodular liver surface or else hepatomegaly, which eventually proceeds down the line to liver atrophy. And you might also see anemia or neutropenia. On a liver biopsy, they're going to describe those periodic acid Schiff positive granules within the hepatocytes. That buzzword, those PAS positive granules, should start to tip you off for glycogen storage diseases. And then finally, a surefire way to lock in that diagnosis would be gene sequencing, or otherwise looking for that enzyme activity, that G6Pase activity. So let's move on now to differential diagnosis. And in my opinion, the thing that you're most likely to confuse this with is Cori disease, which is glycogen storage disease type 3. Remember, Cori disease was a defect in our debranching enzymes. And this does have a similar presentation to von Gierke disease in terms of the liver findings. However, it is a little bit milder because it's not affecting gluconeogenesis as well as glycogenolysis. Those debranching enzymes are all the way over here, sort of away from the gluconeogenesis pathway that was running up glycolysis in reverse. In Cori disease, however, the very helpful thing for you to look at is going to be that serum lactate. In Cori disease, the serum lactate is normal compared to the lactic acidosis that you see in von Gierke disease. Additionally, Cori disease may affect the muscle in addition to the liver. So if you see muscle findings, that's going to start to tip you over towards Cori disease. So maybe weakness or cramping. And on biopsy, either of the liver or the muscle, you're going to see a buildup of limit dextrins. And that just has to do with what enzymes are deficient. And in this case, you're going to see a buildup of that upstream substrate, those limit dextrins. That's a biopsy finding that is specific for Cori disease. Now, Anderson disease, glycogen storage disease type 4, which is a defect in branching enzyme, you could make an argument for that to also be on this differential diagnosis list. It is not frequently tested. We'll talk about that a little more in the video for Anderson disease as for how you can tell the difference between these two. But really sort of the big sticking point I would say for students would be differentiating von Gierke from Cori disease. The treatment for von Gierke disease is going to be oral glucose, and specifically the form they like to give is cornstarch. Cornstarch is a complex carb that's very difficult for the body to digest and break down, so it's like a slow release form of glucose that helps patients avoid those hypoglycemic pits. What you do not want to give are fructose and galactose. And let's go back to the map for a second. And we're at the top of the map here. Fructose metabolism is in dark green. Galactose metabolism is in light green. And look, if you give fructose, that's going to be broken down this way and then try and run up glycolysis in reverse. Again, it's going to get stuck right here. And the same problem with galactose. If you run this all the way through, you're going to just eventually end up making G6P, which is going to cause the same problem. So I've written a little sample vignette here, and I'm just going to go through and highlight the buzzwords for me in this question as I walk through the STEM. All right, so we've got a 5-month-old girl brought to the emergency department after a generalized seizure. She reportedly has been lethargic for the past several weeks. She's at the 15th percentile for height and the 10th percentile for weight. So it looks like she's having some failure to thrive. Physical exam reveals a rounded face. So that's a unique finding, something I would want to clock. In this case, it's the doll-like facies for von Gierke disease, and a distended non-tender abdomen with a liver palpable 4 centimeters below the right costal margin, so hepatomegaly. Lab evaluation is pertinent for serum glucose of 32, so very low, cholesterol of 261, so hyperlipidemia. Uric acid is elevated, and lactate is elevated. So a finding of hypoglycemia is always something you want to pick out straight away, regardless of what the question is asking. That helps to explain our seizure up here. So we've got hypoglycemia and a lactic acidosis in combination with hyperlipidemia and hyperuricemia. And then our other physical exam findings, including those doll-like faces and hepatomegaly. All right, so let's wrap it up. Von Gierke disease is glycogen storage disease type 1. It is from a defect in enzymes that control G6-Pase or glucose 6-phosphatase, which normally breaks down glucose 6-phosphate into glucose. We can either have a deficiency in the expression of the enzyme itself, or else a less common subtype is a deficiency in the G6P translocase protein. Either way, the body is not able to break down G6P, so you get a buildup of that G6P and a decrease in glucose production, both from glycogen storage as well as from a failure of gluconeogenesis. The patient might present with hepatomegaly or renomegaly, and we had hyperlipidemia from sympathetic activation of lipoprotein lipase. Then the big finding we really saw were manifestations of hypoglycemia, for example, seizures. These patients present with a very severe fasting hypoglycemia due to the combined failure of glycogenolysis and gluconeogenesis, and they also experience a lactic acidosis, which is an important finding. You might see gout due to hyperuricemia and some less specific findings like an elevation in liver function tests. Remember, on a biopsy, if you see positive periodic acid shift granules, that should be setting off alarm bells in your head that we might be thinking about a glycogen storage disease of some type. And if we weren't sure which one, specifically if we were confused about Cori disease, remember we were looking at the lactate, which is elevated in von Gierke compared to normal in Cori disease. And these patients were managed with oral glucose, specifically cornstarch, that slow release form, to help prevent the hypoglycemic pits. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on Pompe disease. So we are cracking on down our list of glycogen storage diseases numerically. We've hit glycogen storage disease number 2, which is Pompe disease. Here, an enzyme deficiency is going to cause a defect in glycogenolysis, most notably in the muscle. So remember from our overview video, there were 2 sort of buckets of symptoms we were going to be looking for, either liver symptoms or muscle symptoms. You could have one or the other or both. Here in Pompe disease, we are looking primarily at the muscle. The enzyme that is defective in Pompe disease is this enzyme 9 right here, which was alpha-1,4-glucosidase. And remember, this was only in the lysosome. This was that special pathway where branched glycogen could just have glucose units directly picked off of it instead of having to go through this whole G1P process. It was a convenient little shortcut just in the lysosome. And like I alluded to in the glycogenolysis video, while it's not necessarily a major pathway because it only occurs in the lysosome, in the case of Pompe disease, it becomes critically important. So the enzyme defect is coming from a defective GAA gene on chromosome 17q. A defect in that gene means we're not getting expression of that lysosomal acid alpha-1,4-glucosidase enzyme. Now that alpha-1,4-glucosidase enzyme, the naming is very confusing, but that enzyme will break down both alpha-1,4 and alpha-1,6 bonds in the lysosome. So a defective enzyme is going to result in a decreased hydrolysis of both of those bond types, and it leads to a buildup of glycogen in muscle groups all around the body, but one to really focus on for Pompe disease is cardiac muscle. So in Pompe disease, since we're thinking about muscular symptoms, we're going to be looking at a progression of muscular dysfunction. And the way that this progression is generally going to go is it's going to start in the active voluntary muscles of the extremities. So some muscle weakness in those extremities. Eventually, it will progress to affect the cardiac muscle. And that cardiac muscle manifestation is going to be what they do really like to test. And then eventually that muscle dysfunction will move to the diaphragm. So what does this look like clinically? Well, early on, you might see things like failure to thrive, proximal myopathy, hypotonia, and exercise intolerance, starting with that muscle weakness in the extremities. As things continue and things get worse in the heart, that's when you're going to start to see sort of the crux of this disease here, which is hypertrophic cardiomyopathy. That's really your testable point for Pompe disease. and that's from that glycogen buildup in the cardiac muscle. And so along with that hypertrophic cardiomyopathy, you're going to see cardiomegaly and conduction blocks due to that thick, sort of malfunctioning tissue. And then eventually, as the diaphragm becomes involved, that's when you see respiratory failure, and so unfortunately, these patients often suffer from an early death. One sort of miscellaneous finding that you might see that is by no means pathognomonic, but if you see it in the stem, don't let it confuse you, is macroglossia. So how are you going to diagnose this? Well, on muscle biopsy and cardiac muscle biopsy, what you'll see is PAS positive granules. Okay, remember the PAS positive was an indication of polysaccharides. That's what that periodic acid shift stain stands for. Polysaccharides like glycogen. The presence of PAS positive granules, remember, is not specific for Pompe disease, unless those PAS positive granules, as well as electron-dense deposits, are inside the lysosomes. This is the only enzyme that is specifically only in the lysosomes out of the glycogenesis and glycogenolysis enzymes that we're talking about this chapter. So if the findings on the biopsy are localized to the lysosome, that means you're thinking about Pompe disease. So it's easy to skim over that point when they give you that biopsy finding, but that is going to be supremely helpful for you. Obviously, the very specific way to diagnose this would be newborn screening for the enzyme activity as well as gene sequencing. And you might see some less specific findings like increased creatine kinase indicating muscle dysfunction as well as urinary oligosaccharides. But for Pompe disease, you're not going to see hypoglycemia. Glycogen in the skeletal muscle is only going to be used in the skeletal muscle. All right, so let's talk differential. One of the things that you might confuse Pompe disease for, especially early on in the disease course, are muscular dystrophies. Additionally, muscular dystrophies generally present later. Remember, patients with Pompe disease generally die pretty early. Another disease you could make a case for being on the differential that's not listed here is Cori disease. Remember, Cori disease is a defect in debranching enzymes. And so with Cori disease, there is a possibility of cardiomyopathy, although that doesn't always pop up on test questions. But in Cori disease, there is liver involvement as well as hypoglycemia, which is less likely in Pompe disease. We'll talk more about Cori disease in the next video, so hang tight. So let's talk about management of Pompe disease. Unfortunately, the prognosis is not excellent. However, there is a slightly better prognosis the later on in life that the disease presents. There is enzyme replacement therapy available for that lysosomal acid alpha-1,4-glucosidase, so that's a potential treatment. And then to deal with some of those symptoms of the hypertrophic obstructive cardiomyopathy, you're thinking about pacemaker for conduction defects or even a heart transplantation if the cardiomyopathy gets severe. All right, so let's go through a sample vignette here. So we've got a 5-month-old boy who's presenting to the office with a chief complaint of poor feeding for several weeks. Doesn't tell us a whole lot. Let's keep going. His mother reports difficulty holding his head up to feed, weak suckling, and poor weight gain. Okay, so maybe some sort of muscle weakness. He's at the 20th percentile for height and 5th percentile for weight, so a little bit of failure to thrive. Physical exam reveals hypotonia in all of the limbs and a high-pitched crescendo-decrescendo mid-systolic murmur heard best at the left lower sternal border that increases in intensity when the baby strains. Woof. Okay, that was a lot of information. We're having hypotonia, muscular weakness in the extremities, and they're also describing a murmur, which if you've covered cardiology, you would know that's the murmur for hypertrophic obstructive cardiomyopathy, right? It's high-pitched crescendo-decrescendo. It's systolic. It's at the left lower sternal border. And the murmur with hypertrophic cardiomyopathy increases during the straining phase, like the straining phase of the Valsalva maneuver, because of a decrease in left ventricular volume. So when the baby strains, like when it cries or bears down, that's going to increase the intensity of this murmur. That's one way that they could test you across this biochemistry and cardio thread, is testing your understanding of this murmur. Okay, so we've got the hypertrophic obstructive cardiomyopathy murmur, and we've got the muscular hypotonia. That helps us narrow things down quite a bit. We're already leaning towards Pompe disease. And then when we take a look at chest x-ray, we're going to see cardiomegaly, and a muscle biopsy is going to reveal PAS-positive visible material in enlarged lysosomes. And remember those lysosomes, that was the key there. We can now be very confident that this is Pompe disease. All right, so let's wrap this up. Pompe disease, glycogen storage disease type 2, is a defect in lysosomal acid alpha-1,4-glucosidase expression. This causes impaired glycogenolysis in the muscle and a buildup of glycogen in muscle, and importantly, in the cardiac muscle. The progression of this disease generally goes from the active voluntary muscles in the extremities to the cardiac muscle and eventually to the diaphragm. And the clinical presentation they really like you to know has to do sort of with that second bullet point there, that cardiac muscle involvement. You'll see a hypertrophic cardiomyopathy with some corresponding other features like cardiomegaly or maybe conduction blocks. And to confirm this on biopsy, you're looking for periodic acid shift positive material, specifically in the lysosomes. The enzyme defective in Pompe disease is the only one of the glycogen storage disorder enzymes that we talk about that is sequestered specifically in the lysosomes. And there are a few ways to manage this, at least to mitigate symptoms, like enzyme replacement therapy or some pacemaker or heart transplant options. However, the prognosis is generally pretty poor, and these patients unfortunately suffer oftentimes from an early death. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on Cori disease. So moving on down our list of glycogen storage diseases numerically, we have now reached GSD3, which is Cori disease. And this is going to be an enzyme deficiency in glycogenolysis, leading to impaired glycogenolysis, both in the muscle and in the liver. It means that there are a lot of diagnostics and clinical presentations to cover because we're going to see symptoms both that fall under that liver bucket of symptoms and that muscle bucket of symptoms. But the good news is there aren't that many glycogen storage diseases that have findings across those two categories. So it'll be fairly straightforward to narrow down your differential. So let's get into it. If you remember on our diagram here, there were two enzymes that mediated the transformation of limit dextrins into newly linear glycogen at the very end of glycogenolysis. Those two enzymes were 4-alpha-D glucanotransferase and alpha-1,6-glucosidase, and together those were called debranching enzymes. Cori disease is going to be a deficiency in these debranching enzymes. Now, don't get the debranching enzymes confused with the branching enzyme. All right, the branching enzyme, enzyme 5 over here, that is defective in Anderson disease, which we will talk about next. Branching enzyme, as the name suggests, adds branches to glycogen, as opposed to debranching enzyme, right, which is removing the branch off of the limit dextrin. To keep that straight in your mind, just remember A, B, C, D. Anderson disease is a defect in branching enzyme, that's Anderson branching, and then CD is Cori is a defect in debranching enzyme. So the way that we get that defect in the debranching enzymes is going to be due to a defective AGL gene on chromosome 1p. That means that you're not getting the expression of those debranching enzymes, which remember were 4-alpha-D glucanotransferase and 1-alpha-6-glucosidase, and that leads to a buildup of the limit dextrins upstream of those enzymes. Here though, gluconeogenesis is going to be intact. This enzyme defect is isolated to glycogenolysis. I just bring that up because that will be helpful in explaining some of our diagnostic findings as well as helping to narrow down our differential. So like I said, in Cori disease, we're expecting to see both muscle and liver findings. Some of our muscle findings include failure to thrive, generalized muscle weakness, cramps, and potential cardiomyopathy. However, in a stem for Cori disease, I would say they're actually less likely to bring up this cardiomyopathy when they're talking about Cori disease. It is a possible finding, but often when you are seeing cardiomyopathy in a question stem, you're leaning more towards Pompe disease, and we talked about that differential in that video. But just something to be aware of, when there is cardiomyopathy, you do still have to be thinking about Cori disease. It's still a possibility. However, Cori disease also has liver involvement. So there, the clinical presentation might be hepatomegaly or possible cirrhosis. So let's talk about diagnostics. The big diagnostic picture that you want to keep in mind, other than, okay, I have both muscle and liver involvement that I want to be keeping an eye out for, is on the diagnostics, you're looking for mild fasting hypoglycemia and ketosis with normal blood lactate and normal uric acid. And I'm going to come back to this when I talk about differential diagnoses, specifically the differential diagnosis of von Gierke disease. Put a pin in it for now. The next diagnostic finding is very elevated transaminases. So oftentimes these transaminases might be over 500 units per liter. When you see them over 500 like that, and you're thinking about glycogen storage diseases, that's fairly specific for Cori disease. They might not always give you those values, but if they do, it's a gift and you should take advantage of it. The other liver function marker you might see is an elevated serum biotinidase. Another diagnostic finding you might see is hyperlipidemia. That's due to sympathetic activation of lipoprotein lipase, that sympathetic activation coming from the hypoglycemia. It's the same mechanism to when we talked about von Gierke disease a couple videos ago. Moving on to some muscle findings, findings, one of the first findings you might see is the ischemic forearm test will be positive. So the ischemic forearm test, what they're going to do is they're going to cinch a blood pressure cuff around the patient's upper arm, and then they're going to draw venous blood from the forearm. They're going to take a baseline reading from the forearm, and then they're going to inflate the cuff very high, and they're going to ask the patient to clench their fist and do exercises. Normally, venous lactate is going to increase because you're forcing the patient to do exercise under anaerobic conditions, right? You're cutting off the blood supply to the muscles downstream. That's under normal conditions. But in a patient with a positive ischemic forearm test, like you might see in Cori disease, that venous lactate is not going to increase. And that is an abnormal finding common to Cori disease as well as McArdle disease. That happens because not enough glucose is being produced from glycogen, so lactate levels are not going to rise. During aerobic exercise, skeletal muscle is going to utilize fatty acid beta oxidation for energy. However, when skeletal muscle is working anaerobically, so for example, lifting weights, or in the case of the ischemic forearm test, glycogen is going to be broken down into glucose. And then that glucose is going to enter the anaerobic glycolytic pathway and increase the blood lactate. You can see that on our map here. If you turn glycogen to glucose, that glucose can then run down the pathway here and turn into lactate. But if glycogen is not being broken down into glucose, then obviously it's not going to enter that pathway, and lactate is going to remain low. So that's the ischemic forearm test. That'll come back up in a future video when we talk about McArdle disease. Other muscle findings include an increase in creatine kinase, and then on either a muscle or a liver biopsy, right, it could be either, you'll see the periodic acid shift positive granules that we've been talking about all chapter, signaling the presence of polysaccharides. But unique to Cori disease is you're going to see a buildup specifically of limit dextrin-like structures in the cytosol. Limit dextrin-like meaning either the limit dextrin itself or else that modified limit dextrin. If we just go over to our diagram with the visuals for a second, right, these are the two enzymes that are being cut out in Cori disease. The limit dextrin-like complexes is just talking about either the limit dextrins themselves, which looked something like this, right, with 4 glucose left on the branch, or these modified limit dextrins where some of those glucose from the chain had been added onto the linear strand already, but we still had 1 glucose left on the branch. Just because the two enzymes are involved, you sort of see 2 products here, so they're going to call those 2 together and aggregate the limit dextrin-like complexes. That's specific to Cori disease. And then finally, just to be sure, you can do gene sequencing or an enzyme activity analysis, looking for the debranching enzyme activity. So now let's get into differential diagnosis, and in the von Gierke disease video, we started to talk about this already, how Cori disease is like a milder form of von Gierke disease. So it's going to share the fasting hypoglycemia with hyperlipidemia findings. However, von Gierke disease is more severe. Remember, in von Gierke disease, gluconeogenesis is also defective, whereas we have functional gluconeogenesis in Cori disease. Additionally, in von Gierke disease, very important is to remember that the blood lactate is elevated in von Gierke disease, and it is normal in Cori disease. That's a dead giveaway for you, the difference between those two, as well as the fact that Cori disease involves the muscle and the liver, and von Gierke disease is not going to have any muscle findings. Now, in a question stem, for Cori disease, they don't necessarily have to give you both muscle and liver findings. So that's why it's really important not to just entirely rely on this, but also understand that blood lactate differentiating factor. The other differential we already kind of started to unpack here is McArdle disease, the defect in glycogen phosphorylase. That also is going to have a positive ischemic forearm test, so no increase in blood lactate upon anaerobic exercise. And it is really important to remember that McArdle disease also has this finding because that finding is very heavily associated and tested a lot when they test McArdle disease, so don't forget about it. But McArdle disease only affects the muscle, it does not affect the liver. The last differential I want to talk to you about is kind of a sneaky one, and you're going to have to think all the way back to the galactose metabolism video. This is going to be galactose 1-phosphate uridyl transferase deficiency. Let's go back to our galactose metabolism map. We're talking about enzyme 3 here. One of the themes you should be starting to pick up throughout this chapter is how heavily intertwined galactose metabolism is with glycogenesis and glycogenolysis. So galactose 1-phosphate uridyl transferase right here is the enzyme sort of at the merging point between those two processes. So a defect in that enzyme is going to share the hypotonia and hepatomegaly findings that you see in Cori disease. And that can get confusing because you're thinking, oh, this is something that has both liver and muscle findings, Cori disease. However, you need to be careful and extend your differential beyond just the glycogen storage diseases. In a galactose 1-phosphate uridyl transferase deficiency, however, the key giveaway and the differentiating factor here is really going to be cataracts as well as jaundice. All right, you're not going to see those two things in Cori disease. And the other differences with the galactose 1-phosphate uridyl transferase deficiency is the hypoglycemia is going to be much worse and the presentation is going to be earlier. To manage Cori disease, you're going to give a high protein diet with cornstarch supplementation. Cornstarch, if you remember from the von Gierke disease video, being that sort of slow release form of glucose, we're just giving those things to reduce the pits of fasting hypoglycemia and maintain blood sugar control between meals. All right, let's get into a sample vignette. So we've got a 7-month-old girl brought to the office with a chief complaint of failure to thrive. Her height and weight are at the 10th and 5th percentile respectively. Physical examination reveals hypotonia and hepatomegaly. Right off the bat, even if we freeze right there, we're kind of cheating because we know this is a glycogen storage disorder, but just thinking within the glycogen storage disorders, that's a muscle finding and a liver finding. That narrows it all the way down to either Cori disease or Anderson disease, which we'll talk about in the next video. Let's keep moving. Lab workup is remarkable for elevated urine ketones, serum glucose of 58, so a mild hypoglycemia, not like the 32 we saw in von Gierke disease. Creatine kinase of 100, so elevated, and aminotransferases are both over 500. And then finally, the icing on the cake to really hammer home Cori disease is the lactic acid within reference ranges. The final nail in the coffin against having von Gierke disease on our differential. And then if you got this far along in the question and you still weren't even necessarily thinking about glycogen storage diseases, our next sentence says that a liver biopsy shows a buildup of abnormally structured material within the cytosol of hepatocytes on periodic acid shift stain. Periodic acid shift stain being sort of our buzzword for these glycogen storage disorders. And that abnormally structured material, this is an obnoxious thing that they'll do sometimes. Instead of just telling you limit dextrin-like complexes, they might keep it very vague. Just letting you know, they might not be so nice and give you the buzzword. All right, so let's wrap things up. Cori disease, glycogen storage disease type 3, is a defect in the debranching enzymes that causes a buildup of limit dextrins and limit dextrin-like complexes in the cytosol of muscle and liver cells. As such, you will see muscle and liver findings. The key thing to remember about Cori disease is it looks a lot like von Gierke disease, except the blood lactate is normal, as is the uric acid. Some other good things to remember is that Cori disease is the only glycogen storage disease with transaminases so high that they're above 500. And Cori disease also has a positive ischemic forearm test. We talked about how on biopsy, you might see a buildup of limit dextrin-like structures in the cytosol that stain positive with periodic acid shift stain, which stains polysaccharides. And we talked about a couple of key differentials. So von Gierke shared some of the liver findings, McArdle shared some of the muscle findings, but neither of those two have both. And we talked about the sneaky one that's not a glycogen storage disease, but does have both muscle and liver involvement, being galactose 1-phosphate uridyl transferase deficiency, but that had a big giveaway, which was the cataracts as well as the jaundice. And we manage mostly by managing the hypoglycemia by giving cornstarch and supplementing protein in the diet. That's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on Anderson disease. So moving on down our list of glycogen storage diseases, we have hit glycogen storage disease 4, which is called Anderson disease. Now the unique thing about Anderson disease is it is an enzyme defect just like all of the other glycogen storage diseases, except Anderson disease is the only enzyme defect in glycogenesis. All of the others were glycogenolysis enzymes. So here we're going to see an enzyme deficiency that impairs glycogenesis in both the muscle and the liver. And so we're going to see symptoms in both of those locations, both the muscle and the liver. If you remember from our discussion on Cori disease that we just wrapped up, remember Cori disease or glycogen storage disease type 3 and Anderson disease, glycogen storage disease type 4, are the two most important glycogen storage diseases that affect both of these groups, both the muscle and the liver. The rest of the glycogen glycogen storage diseases that we talk about in this chapter, aside from those two, are either going to affect one or the other primarily. So the defect in Anderson disease, like we said, is a defect in glycogenesis, specifically in enzyme 5 here, which is called branching enzyme. And it's very aptly named, remember, because it adds branches onto linear glycogen to turn it into that branched glycogen for increased glycogen solubility. Remember, we don't want to confuse that with Cori disease, which is a defect in these two enzymes here, which are called debranching enzymes. So you can remember the mnemonic A, B, C, D. Anderson is a defect in branching enzyme, and Cori disease is a defect in debranching enzyme. So just make sure you keep that straight. So we know Anderson disease is a defect in branching enzyme, and the reason that that defect occurs is due to a defective GBE1 gene on chromosome 3p. That's going to cause decreased expression of that branching enzyme, which leads to a decrease in branched glycogen. So let's talk a little bit about the presentation of Anderson disease, and what you are very likely to find frustrating about Anderson disease, or at least I found frustrating about Anderson disease, is none of this is very specific, and there is a huge spectrum of clinical presentations that you can see with Anderson disease. The good news is, in my experience, Anderson disease was more important to be able to understand so that you could identify it as an incorrect answer choice with some of these other glycogen storage diseases. So if you find some of this frustrating and non-specific, don't panic. So in terms of presentation, you're going to really see a continuum of hepatic and neuromuscular subtypes and various presentations associated with each of these. Some things they might mention in the stem would be failure to thrive in early infancy, hepatosplenomegaly and cirrhosis, right? So some liver symptoms. And then for muscle symptoms, you might see hypotonia or cardiomyopathy. You may also see peripheral neuropathy, and that one is a little more unique to some of those neuromuscular subtypes of Anderson disease. The prognosis for this is generally pretty poor, and these patients often suffer from an early death, unfortunately. One thing to make clear here before we move on to the diagnostics is because there is such a variety of hepatic and neuromuscular subtypes within the overarching umbrella of Anderson disease, you may not actually see both muscle and liver findings in every single patient. It's just important to be aware that it can affect both of these organ groups. So let's talk about diagnostics. Because so many of these findings are so all over the place, you're going to lean pretty heavily on this first bullet point here, which is you're looking for the branching enzyme activity or else doing a gene sequencing. Pinpointing the exact enzyme that's deficient or the exact gene that is mutated. That's going to be your best bet here, in all honesty. Again, similar to the other glycogen storage diseases, you're looking for periodic acid shift stain positive granules, either on liver biopsy or muscle biopsy, depending on the subtype. For some of the hepatic subtypes, you can look at liver markers like serum biotinidase, which is produced by the liver, as well as elevated liver function tests like transaminases or alk-phos. If the patient is progressing to liver cirrhosis, you might see a nodular liver surface on ultrasound with hepatomegaly that eventually progresses to atrophy. And then late in the disease course, you might see hypoglycemia. I wouldn't say that this is a hallmark characteristic of this disorder per se, but something that you don't want to let confuse you if it does happen to show up in a stem. So let's talk about differentials here. The first one on your differential might be von Gierke disease, because in the hepatic subtypes of Anderson disease, you might see some similar findings like liver cirrhosis and hepatomegaly. However, a stem for von Gierke disease is going to have a lot of dead giveaway findings, especially in the diagnostics. It's going to have a very severe presentation, and it's going to present with severe fasting hypoglycemia with elevated blood lactate. You'll also see some other findings like hyperlipidemia and hyperuricemia that you're not necessarily seeing with Anderson disease. One differential that's not written on the outline here, but sort of spins off of the von Gierke discussion a little bit, is Cori disease. Like we said, Cori disease also has both muscle and liver findings, and so that might be something that you consider in your differential. However, similar to von Gierke disease, there are very specific diagnostic findings that will help you rule this out pretty quickly. For example, Cori disease, patients may have a positive ischemic forearm test, which we discussed in that video. There's hypoglycemia, albeit milder than von Gierke, in Cori disease that will present earlier on in the disease course than in Anderson disease, as well as hyperlipidemia. And then remember, in Cori disease, you see very elevated transaminases, oftentimes over 500 units per liter. So the frustrating thing with Anderson disease is you don't necessarily have those nice diagnostics to fall back on, but when you do see diagnostic features like this, or the features with von Gierke disease, you should be able to put Anderson disease aside. It's more the absence of these specific diagnostics in a stem that will help you figure out Anderson disease. We can also talk about Pompe disease in our differential discussion here, just because this shares the potential cardiomyopathy that you might see in Anderson disease, as well as the hypotonia, right? Some of those muscle findings. However, in Pompe disease, we were really focused on those hypertrophic obstructive cardiomyopathy symptoms, and Pompe disease does not have any liver involvement. Management for Anderson disease is mostly supportive. Like I was alluding to, this generally has a pretty poor prognosis. Some patients may warrant a liver or a heart transplant, so you can discuss that. All right, so let's go through a vignette. So we have a 6-month-old boy brought to the office with a chief complaint of poor feeding for several weeks. His mother reports difficulty holding his head up to feed, weak suckling, and poor weight gain. He's at the 20th percentile for height and 5th percentile for weight. That all sounds a lot like a question stem we've actually seen before. In fact, it's almost identical to the stem I gave you guys talking about Pompe disease. All we've got right now is muscle weakness and failure to thrive, so we can rule out those diseases that only affect the liver. But other than that, that's about where we're standing right now. So let's keep going. Physical exam reveals hypotonia and hepatomegaly. Now we're talking. We know that there are only 2 of the high-yield glycogen storage diseases that affect both the muscle and the liver. That was Cori disease, and that was Anderson disease. Now we've made some headway. Lab workup reveals a serum blood glucose of 68. We're not shocked by that necessarily. Muscle biopsy reveals the presence of PAS-positive granules. So if we weren't thinking glycogen storage disease, we are now. An enzyme activity analysis reveals decreased activity of an enzyme implicated in glycogen formation. And there's your key. If you think back to our ABCD mnemonic, Anderson was branching enzyme, Cori was debranching enzyme. And of those 2, branching enzyme is the enzyme implicated in glycogen formation. It's involved in glycogenesis, whereas debranching enzyme, right, for Cori disease is glycogenolysis. So let's wrap things up. We've been talking about Anderson disease, which is glycogen storage disease type 4. It is an enzyme defect in branching enzyme, which is a glycogenesis enzyme. So patients with this disorder are not able to produce branched glycogen from linear glycogen. We said the frustrating thing about this disease is we didn't necessarily have any super specific findings or buzzwords that we could really latch onto. There was a variety of hepatic and neuromuscular subtypes with a variety of different findings. So we said the most surefire way to lock in this diagnosis was doing an enzyme activity analysis or else a gene analysis. We talked about how to narrow down some differentials and how if we saw some specific buzzwords for another disorder, we should just stop thinking about Anderson disease, and that was kind of a good way to go about ruling out Anderson disease, which otherwise has a pretty non-specific presentation. And we said this unfortunately has a pretty poor prognosis. Most of the management is supportive. And that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on McArdle disease. So we're working our way down our list of glycogen storage diseases and we have landed on glycogen storage disease 5, which is McArdle disease. Here, an enzyme deficiency is going to lead to impaired glycogenolysis within the muscle specifically. So if we take a look at our diagram of glycogenesis and glycogenolysis, we can see that enzyme 6 here has 2 glycogen storage diseases associated with it. This enzyme is glycogen phosphorylase, which if you remember was the rate-limiting step in glycogenolysis. When glycogen phosphorylase is deficient in the muscle, that is McArdle disease. The alternative, of course, being deficiency of glycogen phosphorylase in the liver. That's an entirely separate disorder called HERS disease, which we'll talk about in the next video. Remember, glycogen phosphorylase was taking G1P units off of the branch of a glycogen polymer, and it kept picking off those G1P units until you ended up with a limit dextrin with 4 glucoses left on that branch. So we know McArdle disease is a defect in the enzyme muscle glycogen phosphorylase specifically. We get that defect from a defective gene called the PYGM gene. That's on chromosome 11q, and just to be aware of the M here is going to stand for muscle, as opposed to HERS disease, which is going to be a PYGL gene defect for liver. A defective gene will lead to a decrease in the expression of muscle glycogen phosphorylase, that rate-limiting step of glycogenolysis. So as a result, you get a buildup of glycogen in the muscle and a decrease in glycogen catabolism. Now, the good news for McArdle disease is the presentation is fairly specific and there are some buzzwords you can look for. The first thing you're looking for is that the patient in the vignette here is generally going to be a bit older than some of the other glycogen storage diseases. Here we're kind of looking for an older child child or an adolescent. And the key buzzwords to take a look for McArdle disease, which I would say is a very high yield glycogen storage disease, so these are buzzwords you're going to want to lock down. You're looking for muscle weakness, cramps, and specifically you're looking for exercise intolerance with a second wind phenomenon. So what that means is that the patient will start exercising and they will very quickly become unable to tolerate it. They'll get cramping, they'll get fatigue. But after about 10-15 minutes of light aerobic exercise, their symptoms will start to improve due to the increased blood flow to the area. That second wind phenomenon is very specific for McArdle disease. You want to look for the second wind or a description of that second wind phenomenon where things get better with gentle aerobic exercise for a couple minutes. That'll really help us when we talk about our differentials. And then some other presentations include rhabdomyolysis from muscle destruction and corresponding myoglobinuria. So you're maybe looking for some darkened urine along with that as well. So let's get into the diagnostics. A diagnostic that they love to put in the vignette with McArdle disease, you see it frequently, is the ischemic forearm test. We talked about this already when we talked about Cori disease, but it is critical to remember that the ischemic forearm test occurs in McArdle disease. As a refresher, the ischemic forearm test involves taking a blood sample from venous blood in the forearm, then wrapping a blood pressure cuff around the upper arm and inflating the cuff very tightly and asking the patient to contract their fist, basically to mimic exercise in anaerobic conditions. So if we go to our metabolism map, right, normally in the muscle under anaerobic conditions, glycogen is getting broken down into glucose, and then that glucose can undergo the anaerobic glycolysis pathway and produce lactate. If we have a defect in glycogenolysis, all of that is not going to occur. So in a normal healthy patient, you're going to see an increase in blood lactate upon that ischemic forearm test. But in a patient where this pathway is defective, that blood lactate is going to stay the same. It is not going to increase upon these anaerobic exercise conditions. So that's what this means by flat venous lactate curve. It's not going to increase from baseline upon inflation of the cuff and subsequent exercise. You'll also see a big spike in ammonia with that anaerobic exercise as well. So that's your big helpful diagnostic finding. There are some other diagnostic findings we can look for as well, like an elevated creatine kinase. These patients may also suffer from electrolyte imbalances that can cause subsequent arrhythmias that you might pick up on EKG. Importantly, these patients will have a normal blood glucose because the liver is not involved. And then, of course, the most helpful diagnostic finding is just figuring out exactly what enzyme or gene is defective. So let's talk about some differentials. The first differentials I want to bring to your attention, even though they're not glycogen storage diseases, are going to be beta oxidation defects and mitochondrial myopathies. So we would need a whole other video to hash out the intricacies of both of these conditions. So the pathophys is beyond the scope of this video here. Both of these present with similar muscular symptoms like the cramping and the exercise intolerance. However, the key here is going to come back to that second wind phenomenon. That second wind phenomenon that they will very likely describe if they're trying to get you to narrow down these differentials is going to be your tip off that it's McArdle disease. You're not going to see the second wind phenomenon with these other two differentials. And additionally, the exercise intolerance takes a little longer to kick in for these differentials. One little thing I'll add for the mitochondrial myopathies as well, just as a little teaser, is frequently in the stem they will give you the buzzword that you see ragged red fibers under the microscope. That's due to compensatory proliferation of the mitochondria in those mitochondrial myopathies. So if you see that description, that is not McArdle disease. Next thing we should discuss on our differential is Pompe disease, another glycogen storage disease that affects the muscle specifically. So patients with Pompe disease also have exercise intolerance and muscle weakness and even arrhythmia. Remember that arrhythmia from the hypertrophic obstructive cardiomyopathy getting in the way of conduction pathways. But in Pompe disease, you're looking for more of those hypertrophic obstructive descriptions and things like cardiomegaly. They'll emphasize a lot of the heart findings in Pompe disease, as opposed to McArdle disease, where you're seeing that arrhythmia from an electrolyte imbalance instead. And then finally, let's bring up Cori disease, glycogen storage disease type 3. The tricky thing with Cori disease, remember, was you could also see those ischemic forearm test findings. And it's very easy when you see that ischemic forearm test to jump right to McArdle disease, but you have to remember to keep Cori on that differential. But the thing about Cori disease is it also affects the liver and will cause mild hypoglycemia. Remember, it looks like a milder version of von Gierke disease with a normal blood lactate. In McArdle disease, we don't have any liver involvement and the blood glucose is normal. So let's talk about the management here, a lot of which is supportive. One of the things that's recommended, though, is a moderate intensity graded aerobic exercise therapy. The goal here is to maintain aerobic fitness and keep those muscles moving in a way that helps maintain the creatine kinase at a low level, right? So that we don't get just muscle atrophy and destruction without exacerbating the symptoms too badly. And that aerobic exercise really is the key here because, as you saw, a lot of our problems were happening with that anaerobic exercise, as evidenced by the ischemic forearm test findings. And you also want to make sure that these patients consume enough glucose before they exercise. So let's do a vignette here. This is a very highly tested disease, and the vignette I'm about to read you is a very classic vignette for this disease. So if you can identify something like this quickly, it'll help you out a lot throughout your prep. So we have a 14-year-old boy. And now, because of the nature of this video, you already know this is a glycogen storage disease. But so I'm going to highlight that because 14 years old is much older than some of the other sample questions we've seen so far in this unit. So that's actually a point for McArdle disease right there already. So we have a 14-year-old boy brought to the office due to several episodes of extreme fatigue and muscle cramping while lifting weights. Lifting weights, remember, being a type of anaerobic exercise. He states that he sometimes feels better with a gentle warm-up and feels much better when he drinks a sugary sports drink before beginning his workout. The gentle warm-up was not just a throwaway line. That's implying the second wind phenomenon. It's sneaky, but just be sure to clock that. It's tough because a lot of times they put a lot of junk in these question stems, but always think about why people might be including things. He also feels better when he consumes a lot of glucose before beginning his workout. All right, let's keep going. He's noticed some occasional darkening of the urine after strenuous activity, myoglobinuria. Physical exam is unremarkable. To confirm the diagnosis, a blood pressure cuff is inflated around his upper arm, and venous blood samples from the forearm are collected at baseline and regular time intervals after exercising the hand. His venous lactate remains unchanged throughout the testing interval. All of this is a description of the ischemic forearm test. Oftentimes they will actually write it out like that and then describe a flat venous lactate curve. So that is a straightforward classic vignette for McArdle disease. All right, so let's wrap things up. McArdle disease, a very important glycogen storage disease to really lock down. This is glycogen storage disease type 5, and it is a defect in muscle glycogen phosphorylase, which is the rate-limiting step of glycogenolysis. Here you're looking for an older child or adolescent who's experiencing muscle weakness, cramping, and exercise intolerance, specifically with that second wind phenomenon. A helpful diagnostic is going to be that ischemic forearm test that we talked about. And we talked about some differentials and how if you can pinpoint exercise intolerance with the second wind phenomenon, you can rule out most of your other differentials. We talked about some management, like a moderate intensity graded aerobic exercise program to maintain muscle health while mitigating symptoms, as well as some glucose consumption before the exercise. And that's all I got for you. Good job. Good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our lesson on HERS disease. So we've reached the end of our new content for this chapter with glycogen storage disease type 6, which is HERS disease. I would say that this is the lowest yield of the 6 glycogen storage diseases that we've talked about. We're going to fly through this lesson pretty quickly, just so that you're familiar with it in the off chance it does come up. HERS disease is an enzyme deficiency that causes an impairment of glycogenolysis, specifically in the liver. If we take a look at our diagram here, we have glycogenolysis, and enzyme 6 here, which is glycogen phosphorylase, breaks down branched glycogen by pulling off G1P units until we reach the form of that glycogen called a limit dextrin. Now, in the last video, we just finished talking about McArdle disease, which was a deficiency in glycogen phosphorylase in the muscle. However, when this enzyme is deficient in the liver instead, that is called HERS disease. And remember, this was the rate-limiting step of glycogenolysis. So we know HERS disease is a defect in expression of hepatic glycogen phosphorylase, and we get that defect from a defective PGYL gene on chromosome 14q. Remember, the L here is going to stand for liver, as opposed to the M in the PGYM gene, which stood for muscle, which was the gene defect that we saw in McArdle disease. So a defect in that rate-limiting step is obviously going to cause a buildup of glycogen in the liver, like we've seen with so many of our other glycogen storage diseases, and and a decrease in glycogen catabolism. The presentation for HERS disease is pretty non-specific. Like we said, we are focused in the liver, so we're thinking about some liver symptoms like we talked about in our overview video. None of these are tremendously specific for HERS disease, but they include hepatomegaly, hepatic fibrosis, and then if this disease is poorly controlled, it can lead to other problems like a short stature, delayed puberty, osteopenia, and osteoporosis. You'll see that in more severe presentations of the disease. Let's talk about some diagnostics. So in HERS disease, you might see mild hypoglycemia with ketosis, but intact gluconeogenesis. And that intact gluconeogenesis is why that hypoglycemia is mild. And if that's giving you deja vu, that's because that's a very similar description to what we talked about with Cori disease. So we'll get into our differential discussion in a minute here. Some other findings that are also not terribly specific include liver markers like elevated transaminases and a decrease in prealbumin. You'll also see hyperlipidemia here. The diagnostic ultrasound finding that corresponds to some of these presentations is hepatomegaly with diffuse echogenicity. And then of course, like all of the other glycogen storage disease we've talked about, a big, bell ringer phrase that you might see to indicate that you should be thinking about glycogen storage diseases, although not specifically this one necessarily, are periodic acid shift positive granules, in this case on a liver biopsy. And then of course, the most specific and definitive way to diagnose would be gene sequencing and an enzyme activity analysis. So if all that didn't feel very new or specific or helpful, I don't blame you. HERS disease in general is very non-specific, kind of a similar problem like we had to Anderson disease. So let's focus on the differentials. You're thinking about other disorders that are really affecting the liver. Your highest yield ones that you're going to want to worry about are von Gierke disease and Cori disease. You could make a case for putting hepatic subtypes of Anderson disease on this list as well. However, HERS disease and Anderson disease are two of the lowest yield glycogen storage diseases, and I would say it's less likely that you would have to differentiate between those two without a very clear and specific finding like an enzyme activity analysis. With the two listed here, it can get a little trickier though. So von Gierke disease is going to share a couple of the findings like the hypoglycemia as well as the hyperlipidemia. However, like we emphasized, the hypoglycemia in HERS disease is mild. In von Gierke disease, this is tremendously severe, and you're also going to see an elevated blood lactate along with that hypoglycemia, as well as potentially hyperuricemia. So remember, all of those findings together were a pretty clear tip-off for von Gierke disease. HERS disease doesn't have any very helpful findings like that. The one you might be a little more worried about figuring out is Cori disease, because Cori disease did have that mild hypoglycemia with intact gluconeogenesis, as well as hyperlipidemia. It even had elevated transaminase. However, the transaminases for Cori disease were generally very, very high, like greater than 500 units per liter. You're less likely to see something that dramatic in HERS disease. Additionally, Cori disease, remember, didn't only affect the liver, it could also affect the muscle. So if you see muscle findings in the stem, like muscle weakness or what have you, you're not thinking about HERS disease. HERS disease is a glycogen phosphorylase deficiency localized to liver glycogen phosphorylase. The management here is primarily supportive, and you can supplement cornstarch and protein like you can do for a lot of these glycogen storage diseases that affect the liver to prevent those pits of hypoglycemia. I'll show you a quick vignette, and you'll notice very quickly there's not a whole lot they can give you in the vignette here. So you're kind of almost looking for what's missing, right? What findings for the other glycogen storage diseases do we not have here? Things like hyperuricemia, elevated blood lactate, maybe muscle findings. The absence of all of those can be just as helpful as what's in the vignette. So we've got an 11-month-old girl brought to the office with a chief complaint of abdominal distension. Physical exam reveals a palpable liver 3 centimeters below the costal margin, so hepatomegaly and abdominal distension. Lab workup is remarkable for a serum glucose of 64, so maybe a little bit low. Total cholesterol of 254, so that's high. Elevated transaminases. That's helpful, but not super helpful because they didn't give us the value, so that doesn't help us with Cori disease at all, but something to be mindful of nonetheless. And a prealbumin level of 11, which is low. And so like I said, HERS disease makes for a not tremendously helpful vignette. Maybe they would even give you an enzyme analysis here and just straight up tell you, and they might tell you about periodic acid shift positive granules if they want to tip you more towards glycogen storage diseases, but you're getting the idea. So it's a little unsatisfying, but that's how it goes. glucose. So let's wrap things up. Our final glycogen storage disease that we are talking about is HERS disease, which is glycogen storage disease type 6. It's an enzyme defect in hepatic glycogen phosphorylase, which is encoded by the PGYL gene, which leads to impaired glycogenolysis. Specifically, this is impacting the liver, so we're going to see liver symptoms like hepatomegaly, fibrosis, and we'll see some diagnostic findings similar to ones that we've seen with other glycogen storage diseases like mild hypoglycemia with ketosis and intact gluconeogenesis, elevated liver function tests, hyperlipidemia. The most surefire way to just lock in that diagnosis would be a gene sequencing or an enzyme activity analysis. But if not, we talked about some characteristic features of two of our major differentials, which were von Gierke disease and Cori disease, and how the absence of some of the buzzwords associated with those two diseases in a question stem can be pretty telling and helpful to you for moving those down your differential. And the management here was primarily supportive. All right, you did it. No more new glycogen storage diseases for you. We're going to wrap everything up in the next video. Till then, that's all I got for you. Good job, good luck, and keep practicing. Hi everyone, my name is Paige and thank you so much for joining me for our summary lesson on the glycogen storage diseases. There's no new material in this video if you've been following this chapter along chronologically. We're just going to sum everything up in case your exam is tomorrow and you need all this information in one place. For each disease, we're going to go through the location of which tissues are being affected by the enzyme deficiency, what the defective enzyme is exactly, the function of that enzyme, and then some clinical presentation and diagnostic information. It's not going to be all-encompassing, but it's going to help you identify those key buzzwords to get through questions quickly and accurately. We're going to go down our list numerically from 1 to 6, starting with glycogen storage disease type 1, which is von Gierke disease. This is affecting the liver, and the defective enzyme here was G6-PAse, right, glucose 6-phosphatase, which was in charge of converting glucose 6-phosphate into glucose. And this was pretty unique compared to the rest of the other glycogen storage diseases because this impacted not only glycogenolysis, but also gluconeogenesis. So if we take a look at our diagram over here, von Gierke disease was a deficiency in this enzyme 10 right here, glucose 6-phosphatase. So after those glucose 1-phosphate units were cleaved off of the end of glycogen branches, that phosphate group was shuffled around from the 1 position to the 6 position, creating glucose 6-phosphate. And then that phosphate ultimately had to be cleaved off of G6P to just turn it into normal unphosphorylated glucose. The enzyme that mediated that was that glucose 6-phosphatase. So we had defective glycogenolysis, but then also, if you remember, or you can see on the diagram here, gluconeogenesis is being represented by these blue arrows running up glycolysis in reverse. And the last step of that gluconeogenesis also involved that glucose 6-phosphatase enzyme. So because both glycogenolysis and gluconeogenesis were being affected, we ultimately had a pretty severe presentation that we were worried about with von Gierke disease. And these symptoms were all surrounding the liver because that's where this pathology is taking place. So we saw hepatomegaly and renomegaly. And then the high yield point to take away from von Gierke disease that you're looking for in a question stem is fasting hypoglycemia with mild ketosis and elevated blood lactate. If that is the line that you can remember about von Gierke disease, you're going to be in pretty good shape. And that elevated blood lactate is going to help you differentiate von Gierke disease from things like Cori disease and HERS disease, which has normal lactate as well. Other things you might see in von Gierke disease include elevated triglycerides and hyperuricemia, and some things that come along with that, like maybe dull-like faces with the high lipid content in the body, and the corresponding clinical presentations with those diagnostic findings. For example, gout with hyperuricemia. Let's move on now to Pompe disease, glycogen storage disease type 2. This is an enzyme defect primarily in the muscle, and it's a defect in lysosomal acid alpha-1,4-glucosidase. Back to our diagram here, remember we had a minor glycogenolysis pathway that happened only in the lysosome. That was mediated by enzyme 9, lysosomal acid alpha-1,4-glucosidase. And a defect in that enzyme was Pompe disease. So we had defective glycogenolysis within the lysosomes of the muscle. Pompe disease is another highly tested glycogen storage disease on the spectrum of testability here. And the big thing you're going to want to remember is that it causes cardiomyopathy and conduction blocks, specifically a hypertrophic cardiomyopathy. So while Pompe disease is not the only glycogen storage disease with the potential for cardiomyopathy, it should be very high up on your differential when you see obstructive cardiomyopathy, cardiomegaly, things like that. You can remember Pompe affects the pump, being the heart. But it doesn't just affect the heart, it affects other muscles as well. So you might see proximal myopathy and hypotonia, which eventually will lead to respiratory insufficiency as it progressively affects the diaphragm. You might also see macroglossia, kind of a strange little finding there. And specifically, you'll see periodic acid shift positive granules within lysosomes. It's going to be specific to the lysosomes. This is the only glycogen storage disease of the 6 that has that specificity. Let's move on down to Cori disease. And Cori disease affects the muscle and the liver, and it is a defect in the debranching enzymes. The debranching enzymes are actually 2 enzymes that you can kind of think of as a unit for the sake of glycogen storage diseases. Often they'll be referred to just as debranching enzymes instead of their birth names here, alpha-1,6-glucosidase and 4-alpha-D-glucanotransferase. These enzymes are also involved in glycogenolysis. So let's take a look at our diagram. Enzymes 7 and 8 here together are the debranching enzymes. And you can see here, collectively, those debranching enzymes are what mediate the transformation from limit dextrin back into linear glycogen. It finishes cleaving that branch. So Cori disease is going to look a lot like von Gierke disease from a diagnostic standpoint, but it's going to be milder. The reason that it is milder is because gluconeogenesis is intact. Additionally, lactate in the blood is going to be normal. The other difference from von Gierke disease is going to be the potential for muscle involvement as well, right? Remember, von Gierke disease was only the liver. Cori's the muscle and the liver. So you might see muscle weakness or cardiomyopathy. You'll also see very elevated transaminases to the tune of more than 500 units per liter. They won't always give you the values in the stem, but if they do, that's a great tip-off because Cori disease is the only one where you're going to really see it spike up above 500 like that. And the other thing they might describe are cytosolic limit dextrin-like complexes on a biopsy. That's because the debranching enzymes are what break down the limit dextrins and those sort of modified limit dextrins into newly linear glycogens. Keep on moving down the line here to Anderson disease, one of our lower yield diseases, but one that's still important to be aware of. This is going to affect the muscle and the liver, but there are a variety of subtypes, so it may affect one or both. The defective enzyme here was branching enzyme, and the unique thing about branching enzyme is it is the only enzyme deficiency within these glycogen storage diseases we've been talking about that is a glycogenesis enzyme. So let's check out our diagram. Enzyme 5 here is branching enzyme, and that is what is defective in Anderson disease. And as you can tell, branching enzyme is involved in glycogenesis. It takes the linear glycogen and adds branches onto it, as the name suggests, and turns it into branched glycogen, which is that soluble storage form. The findings here, if you remember from the video, were frustratingly nonspecific. We could see some hepatosplenomegaly and failure to thrive. So those are some liver symptoms, along with cirrhosis as another liver symptom. From the muscle symptom side of things, we might see hypotonia, muscle weakness, and cardiomyopathy, as well as peripheral neuropathy and some of those neuromuscular subtypes. None of those in isolation is necessarily a dead ringer giveaway for Anderson disease, but those findings together can help you narrow down that differential at least a little bit. McArdle disease was glycogen storage disease type 5, this one affecting the muscle only. And this was a defect in muscle glycogen phosphorylase, which was a glycogenolysis enzyme, specifically the rate-limiting enzyme of glycogenolysis. If we take a look at the diagram here, enzyme 6 has 2 glycogen storage diseases associated with it, enzyme 6 of course being glycogen phosphorylase. When it is the muscle form of glycogen phosphorylase that is deficient, that is McArdle disease. That's a failure of muscle phosphorylase to pluck those G1P units off of the branched glycogen to form those limit dextrins. Here with McArdle disease, we had some very specific findings that were dead ringer giveaways for McArdle disease, specifically muscle weakness and exercise intolerance with a second wind phenomenon. That second wind phenomenon being improvement of symptoms after about 10 to 15 minutes of gentle aerobic exercise. We might also see rhabdomyolysis and myoglobinuria. Here on the labs, we saw normal blood glucose, right, because we didn't have liver involvement, and a flat venous lactate curve with an elevation of ammonia. And I'll add here that this is on the ischemic forearm test, and that was mimicking anaerobic exercise conditions. So really that second wind phenomenon is what you want to keep a major eye out for. That's very specific for McArdle disease. And that's an improvement of symptoms with an increase in blood flow. And then the last glycogen storage disease we talked about was HERS disease. disease, which is a defect in the liver of liver glycogen phosphorylase. Again, just like with McArdle disease, this is a glycogenolysis enzyme. The only difference here, if you take it to the diagram, it's that same glycogen phosphorylase, that rate-limiting enzyme of glycogenolysis, but now it is specifically in the liver instead of the muscle. This caused some very non-specific findings like hepatomegaly, fasting hypoglycemia and ketosis, and hyperlipidemia, and like we mentioned, a normal blood lactate. And we could also look for an enzyme deficiency or a gene sequencing analysis as well to help us lock down this diagnosis. One thing that all of these had in common was the biopsy finding of periodic acid Schiff stain positive granules. And depending on which tissue it was affecting, that would affect the biopsy site. But the periodic acid Schiff stain stands for polysaccharides and should tip you towards a potential glycogen storage disease diagnosis. In my eyes, the 3 glycogen storage diseases you do want to really have locked down are von Gierke disease, Pompe disease, and McArdle disease. If you can remember these high-yield lines with these specific findings, you'll be in a really great place to tackle these questions and narrow down these differentials. Start by figuring out, okay, is this patient having a defect in the muscle, the liver, or both? From there, you can hack away at your differentials and then start looking for specific findings and go from there. So that's all I have for you for this chapter. If you have any questions, comments, concerns, problems, troubles, worries, hopes, dreams, fears, send them all my way. Otherwise, good job, good luck, and keep practicing. All right, go ahead, take a minute to read through the question, and then we'll come back together to talk about the answer. All right, so we'll read through the question in just a minute, but I'm just going to give you my honest test-taking strategy here. Typically, when I see a big question with a lot of parts like this, I'll actually jump right to the last sentence to see what the question is actually asking. It says, this patient most likely has a deficiency in an enzyme responsible for which of the following reactions? A, B, C, D, E. So it's referring to this diagram here. And we were only really given the names of the reactants and the intermediates. We don't have the names of any enzymes. So before I go through and read the question, I might as well identify these enzymes, since I'm going to have to do it anyway in this question, it seems like, and use that to establish my differential, so that going into this question, I know sort of how to direct my thoughts. So it looks like there are 3 big pathways here outlined on this diagram. Glycolysis is over here on the left. We're looking at glucose turning into pyruvate. Glycogenesis is here in the middle. We're seeing how those glycolysis intermediates turn into glycogen. And then glycogenolysis is on the right. We can see how we debranch and eventually break down glycogen. Enzyme A here is glucose 6-phosphatase. That is the enzyme that is defective in von Gierke disease, so answer choice A is going to be representative of von Gierke disease. Enzyme B is alpha-1,4-glucosidase, which is defective in Pompe disease. Enzyme C is branching enzyme, which is defective in Anderson disease. Enzyme D is glycogen phosphorylase, and that is actually defective in 2 different disorders of glycogen metabolism. When it's defective in the muscle, that's McArdle disease, and when it's defective in the liver, that is Her's disease. And then E here is actually representing sort of 2 different reactions. Together, the enzymes that it's representing are called the debranching enzymes, and those debranching enzymes are defective in Cori disease. Great, so we have our differential, and now we can go into this question knowing that it's gonna be one of these 6 things. So we have a 10-month-old girl brought into the pediatrician because of a 6-week history of poor feeding and irritability. It looks like she's having some failure to thrive. She's low in her height curve, but she's very low on her weight curve. Physical exam is revealing poor muscle tone and hepatomegaly without splenomegaly. We have fasting labs, which they made a point to emphasize, and let's take a look at them. Glucose here is low. AST and ALT are both very, very high. They're up in the 500s. Creatine kinase is also high. Lactate, though, within normal limits. And our urine dipstick looks totally normal, except for a small amount of ketones in the urine. And so to wrap up the question, the physician is suspecting a defect in a gene encoded by chromosome 1p. Now, if you memorized all the chromosomes that all of the genes that these enzymes are on all the more power to you. I bet this was a very easy question. I can guarantee you, though, I did not know those for my step exam. And that's okay. We don't need to remember that to be able to solve this question. Right away, what is jumping out at me? This fasting hypoglycemia and these very elevated transaminases. So when I think through the glycogen storage diseases, which all of these are, I'm thinking of two big categories of symptoms. There's involvement of the muscle and involvement of the liver. Pompe and McArdle disease typically have predominantly muscle findings. Von Gierke and Her's disease only have liver findings, and Cori and Anderson have both muscle and liver findings. Sure enough, in this patient, we have both muscle and liver findings. We have poor muscle tone, we have hepatomegaly, we have very elevated LFTs, and we have an elevated creatine kinase, which is a sort of muscle lab. So right off the bat, I'm thinking of either Cori or Anderson disease. In this case, these LFTs are jumping right out at me. LFTs up greater than 500 is very unique to Cori disease among the glycogen storage diseases. The other thing that points me towards Cori disease and away from Anderson disease is this mild fasting hypoglycemia, as well as the small amount of urine ketones. All of those features point me right towards Cori disease, represented by answer choice E. So let's go through our incorrect answers, the first one being von Gierke disease. In von Gierke disease, you do see fasting hypoglycemia. Typically, it's a little more severe than in this case. You do also see mild ketosis. However, 1, the involvement of muscle findings, and 2, this normal lactate are what tell you that this is not von Gierke disease. Von Gierke disease often presents with a lactic acidosis. And that's because when we knock out glucose 6-phosphatase, we get a buildup of glucose 6-phosphate, and that has downstream effects on the rate-limiting step of glycolysis. We end up increasing our anaerobic metabolism, creating more lactate. There's no lactic acidosis in Cori disease, and like we talked about in the video on Cori disease, you would see inappropriately normal lactate levels in an ischemic forearm test. Also, like I said, these very high transaminases are pretty unique for Cori disease. I wouldn't really expect to see them this high in von Gierke disease. So von Gierke is out. What about Pompe disease? Pompe disease is a very different clinical presentation. Typically, that's characterized by cardiomyopathy without these ultra-high transaminases. It's primarily muscular symptoms, so the elevated CK supports this, but nothing really else, so Pompe disease is out. What about Anderson disease? This was maybe our most tempting because Anderson disease also presents with primary muscle and liver findings. Early on in the course of Anderson disease, though, we wouldn't really expect to see hypoglycemia. Not unless you've already progressed to Anderson liver failure, and even in that case, we would not expect these transaminases to be that high. CK can be normal or elevated in Anderson disease, so it's a little hard to make a judgment call based on that point, but also these urine ketones we wouldn't really expect to see in Anderson disease. Now, choice D represents either McArdle disease in the muscle or Her's disease in the liver. In McArdle disease, I would not expect to see the liver findings. Instead, I'd be looking for an older child with muscle weakness and exercise intolerance with a second wind phenomenon. So the more they exercise, they actually get some of that strength back. You wouldn't expect hypoglycemia or ketosis, nor would you expect to see those transaminases. The other thing is McArdle disease is characterized by myoglobinuria, or myoglobin from broken down muscle in the urine. Myoglobinuria on a urine dipstick would show positive blood. And if they gave you a urine microscopy, you would expect to see positive blood with no red blood cells. That's classic for myoglobinuria. And Her's disease, very uncommonly tested. It does involve the liver, but we still wouldn't expect those transaminases to be that high, and we wouldn't expect those muscle findings. So, in summary, Cori disease is a glycogen storage disease characterized by a defect in the debranching enzymes. It's characterized by impaired glycogenolysis affecting both the muscle and the liver. It's characterized by mild fasting hypoglycemia, very elevated transaminases, sometimes greater than 500, mild ketosis, a normal blood lactate, and an elevated creatine kinase. Cori disease is a defect in the debranching enzymes that help turn limit dextrins into newly linear glycogen in the process of glycogenolysis. And that's all I got for you. Good job, good luck, and keep practicing.